Fernando, it's welcome and it's great to have you with us. So please start.
Okay, I work at EMBRAPA Brazil sorghum. First, thanks for the invitation to be talking tonight, and I'll try to pass some experiences that we have been for the last 4 decades. And I work at EMBRAPA Brazil sorghum in the southeast of Brazil, that it's located in Sete Lagoas, Minas Gerais state. Next, please. So Brazil is a tropical country, and we have in many regions like 3 crops a year. And this situation creates a green bridge among these crops. And fall armyworm may attack more than 180 crops and survive and reproduce from peanuts to grasses and everything. So we have a huge production, but at the same time, a huge problem with many insect pests. Next. So we work mostly with fall armyworm, Spodoptera eridania, Spodoptera cosmioides, and in cotton and soybeans also soybean looper. And it's very easy to identify fall armyworm with this inverted Y in the forehead. And it's very clear here in the first picture. Next, please. Well, this is not what I sent you. Okay, fall armyworm. So it's a very important pest in Brazil. This is the main characteristic of the damage in the field. And this is the place where it used to have a treatment sprayed with water, so it may eat everything from all leaves and all the plant. Next, please. This we can see the inverted Y in the forehead that it's the major characteristic to differentiate when it attacks the corn plant in the bottom. Next, please. The life cycle is around 30 days. It's a temperature dependent. So in the lab, we get about the same as in the field. And it's easy to rear, but it's really hard to control. Next, please. For the fall armyworm management, you can use chemical control, cultural control to expose like pupa to high temperatures, damage pupa during plowing, biological control. People use trichogramma and some pathogens like baculovirus and Bacillus thuringiensis and some fungi. And of course, transgenic maize. We recommend different genes. Protein is expressed in this maize. And I'm going to just talk briefly about the maize that we developed at EMBRAPA Maize and Sorghum, the first Brazilian transgenic maize against fall armyworm. So we have, for the biological control, the parasitoids, they rear like, it's used trichogramma, Cotesia, and some others. Some predators like ladybugs, sucking bugs, and lacewings. Like the capital of the state is about 4 million people, and they distribute ladybugs and lacewings homes. And we work, my area of work is mainly baculovirus and Bacillus thuringiensis, but it's also in the market Wolferia and Metarhizium. Next, please. Baculovirus, it's efficient. I'm gonna show in the end some of the products that we have developed. It's you have to use healthy caterpillars to multiply this agent. It's you cannot use fermentation system so far. Bacillus thuringiensis, we can use rice as substrate like solid fermentation. We have a project in Africa, and in Brazil, they use mostly large-scale fermentation systems like liquid fermentation. I'm going I'm going to show you some pictures. Trichogramma is an egg parasite, rear the host and the infect and distribute in the field. Pheromone is just for monitoring the presence of adults. And fungi, as I said, both Bacteria and Metarhizium. Next, please. Transgenic maize. So we have the first transgenic Brazilian maize with CRY1DA gene from our Bacillus thuringiensis collection. It's very effective against fall armyworm. It was developed at EMBRAPA based on sorghum and Helix, a Brazilian private company. Now it's beginning to some commercial trades with China and Europe. Next, please. So for successful application of biological products, timing is the most important. So the product application time, it's after 4 p.m. or if you have a formulation with UV protectant. The spray should be between 100 and 120 liters per hectare, and the pH, the water pH about 6 to 7. This is because you have to, Bt and baculovirus must be ingested, so you have to get a very reasonable amount of water when you spray. And spray 1 week after plant emergency. We are having, like in Brazil, heat waves last year, this year. So if you wait too much, you just have overlapping sizes, and it's a really big problem. Next, please. I'll try to talk a little bit about biological control with baculovirus from the perspective of what we got so far. Next, please. So we have a baculovirus collection for soybean looper, the first one. We have many isolates for fall armyworm, that it's the pink one, and in the middle one. And we have also for Helicoverpa armigera and Helicoverpa zea, the last two hanging on the artificial diet. So this baculovirus collection, we get those in the field. And we study and sequence most of them, like the DNA. Next, please. For baculovirus, for fall armyworm, we have mostly, EMBRAPA has mostly negotiated with private companies, isolate number 6 and 910, and they are very different. Isolate number 6 does not disrupt the integument of the dead insect immediately after death, and the 19 disrupts the integument. Both were sequenced. Next, please. We also try different types of formulations for our final product, and we also have a wettable powder, 50 grams per hectare, with a certain amount of PIBs, polyhedra, per hectare, and this I can explain later how we do that, but we try different formulations in our lab too. Next, please. Biological control with Bacillus thuringiensis, that it's a Bt with that they have a spore and they develop a crystal that it's toxic to insects, many insect orders, different orders. Next, please. So we have a Bt collection that we collected about 2,000 samples of soil, grain dust, and from different Brazilian states. And we have today more than 4,600 isolates. And they are kept in different buildings and freezers, -80 and -20. Next, please. We also have a soil collection at EMBRAPA maize and sorghum that we use to get some other type of fungi and bacteria like Bacillus subtilis, amylo, and some others. Next, please. We tested all Bt isolates against some specific insects, like fall armyworm. The best isolates were negotiated by EMBRAPA with private companies for large-scale research and development. Private companies signed a contract with EMBRAPA, developed their own formulation, registered, and placed it on the market. EMBRAPA also develops with our lab our own formulations, IPM, and biological control field trials, and monthly survey of FAW armyworm. Next, please. So we have with BT the isolation, identification, bioassay against the insect pest, the target, pilot scale fermentation system, and we transfer that to the private companies with large scale fermentation systems. Next, please. So this is, we are trying new equipments. These huge bags are, you can grow up to 4 strains at the same time. They are graphene plastic bags, and they are sterile, so you can control with a computer in the mid, and they do not contaminate, and everything is sterile. So we are going to start that on March 25, like next year. Next, please. We have a project with New Zealand and develop New Zealand's, the name of the project is Developing New Zealand's Climate Resilient Integrated Pest Management Strategy Against FAW armyworm. So it's going to start in January 25, and it's a bioprotection, biocontrol, the New Zealand Institute for Plant and Food Research Limited. So they got in touch, we start talking, and we approved this project, and we are going to New Zealand next March, and they are coming to visit us also March. Next, please. You're going to see Argentina, and it's a 2-year project, and they are gonna we are gonna try to exchange knowledge about IPM practices, including biological control for farm armyworm, and they will like enable us. This is from the project to develop a climate resilient IPM program against this invasive pest, and tailored for specific for New Zealand. So we are they are going to come to visit us, and Argentina. We are the major. They just stated that our laboratory at EMBRAPA has all the necessary resources and tries to provide them some knowledge about biological pesticides. We have about 13 biological pesticides developed in our lab, that it's a biological control laboratory at EMBRAPA Brazil sorghum, and they are registered to date, as well as access to field stations for IPM trials. Next, please. So we have this product. I just try to do this table with the products that we developed in our lab and the pests in the mid and the company. So Acera is for 2 strains of bacillus for Spodoptera and soybean looper, and developed by Balagro. Crystal is for Spodoptera, and they are going to increase the pest. So by Lalimand, former Farroupilha in Brazil. Cartuchovite and Baclonate and Destroyer, Virmix, Spodovir Plus, Vir Control. They also have clones to other companies. They are all from, except for Spodovir Andermatt from Switzerland, and they are produced in Canada. And we also have for Chrysodaxis, Vir Control CI and Vir Control HI, that it's Silicover Parmitra, all developed by Symbiosis in Brazil. Next. Thanks for your attention.
With Joe. Thanks, Joe. And the people that have helped in the background, Zatamel and Leandra today on the chat, and those that have joined us, Ashley and Heidi, and even Tech. So thank you, and thank you, Callum. And that's an end, and we will see you soon. Thank you. Bye.
Bye now.
Bye. Thank you.
Bye.
Thanks.